No issues. Let me move forward with this. So this session is I will be divided into the four parts. Where in the initial part of the session, I'll be providing the summary of the previously lectures. That is, this since this is dedicated to live session for week one, I'll be providing a summary to the week one lectures. Second, I'll be using some sample problems where the sample problems will be displayed to you using slides and Google Meet polls. You can answer the questions later. The explanation of the problem will be provided. Later, if the time permits, I'll discuss the questions that are posted in the Swayam portal or the NPTEL portal. And finally, I'll take any doubts from you. So before moving forward, I have few questions for you just to understand the target audience standard and at what level of education they are. So kindly check the polls for the second question as well as the third question and answer them. Okay, thanks for your responses. Let me move forward. So I'll be starting with the summary. So in summary, I'll be discussing with the lecture 1A. So this lecture provides an overview of bioinformatics, different aspects of bioinformatics and application of bioinformatics. So before discussing, I'll start with what is bioinformatics. Bioinformatics is an interdisciplinary field that develops methods and softwares for understanding biological data, in particular when the data sets are large and complex. As an interdisciplinary field of science, bioinformatics combines biology, chemistry, computer science, information technology, mathematics, and some statistical knowledge to analyze and interpret the biological data. Okay, now here we have understood that bioinformatics has something to do with biological data, and we all also know that biological experiments can generate large amounts of data. Here, so here the role of the bioinformatics is about acquiring, managing, analyzing, and understanding these biological data. This bioinformatics field has been used for in silico analysis of biological queries using computational and statistical techniques. Bioinformatics include uh, biological studies that use computer programming as part of their methodology, as well as specific analysis pipelines that are repeatedly used, particularly in the field of genomics. Common uses of bioinformatics include identification of candidate genes or single nucleotide polymorphism. Often, such identification is made with an aim to better understand the genetic basis of a disease, their unique adaptations, and the desirable properties or differences between the disease populations. Bioinformatics also tries to understand the organizational principles within the nucleic acid and protein sequences. This is the major concept of will be in our discussion today. So coming to the what are the aims of bioinformatics is the major aim of bioinformatics are development of databases containing all biological information, development of better tools for data designing, annotation and mining, design and development of drugs by using simulation software or any other softwares, design and development of software tools for structure prediction function, annotation and docking analysis, Finally, creation and development of softwares to improve tools for analyzing sequences for their function and similarity with other sequences. Lecture 1b is about complexity of biological systems. If you talk about complexity of biological systems, we have different ways. First, we start with the DNA nucleotide sequence. It is started with the order of different nucleotide bases, that is arrangement of different nucleotide bases. In DNA in general, we have 4 nucleotide bases there, A, T, G, C. So a systematic arrangement or an orderly arrangement of these leads to a nucleotide sequence. Once we have nucleotide sequence, then we go for the nucleotide structure, and from nucleotide structure then we go with the gene expression. That is, here we are going with DNA to RNA and RNA to protein. Finally we get the protein sequence, and once we had the protein sequence, then we go with the next level. This is called protein structure. Then from protein structure we can get several information such as protein interactions, and protein interactions are the basis for the cell signaling and etc. So this is the broadly the complexity of biological system. So what is the role of bioinformatics in understanding these complexities? In order to understand these, the field of bioinformatics has evolved such that most pressing tasks now involve the analysis and interpretation of various types of data. This also includes nucleotide and amino acid sequence, protein domains, and protein structures. The important subdisciplines within bioinformatics include development and implementation of computer programs that enable efficient access and management and use of various types of information. Second one is development of new algorithms that could be mathematical formulas and statistical measures that assess the relationship among the members of large dataset. For example, there are methods to locate a gene within a sequence, to predict the protein structure or function, and to cluster the protein sequence into families or related sequences. Coming to the primary goal of bioinformatics is to increase the understanding of biological processes. What sets it apart from other approaches is its focus on developing and applying computationally intensive techniques to achieve this goal. The major research efforts in the field of bioinformatics include sequence alignments, gene finding, genome assembly, drug discovery, protein structure alignment, protein structure prediction. Of course, protein structure prediction would also be including the prediction of gene expression, protein-protein interactions, and recently we know genome-wide association studies and modeling, which helps in the modeling the evaluation and other cell division models. Currently, bioinformatics entails the creation and advancement of databases. So where day-to-day life of a bioinformatician will start with a database, algorithms, and computational statistical techniques. And now there is a theory to solve the formal and practical problems arising also. Coming to the second lecture, this lecture is about sequence analysis in bioinformatics. Okay, before I move into that, I see some people recently joined. Can I have a hand raise of the people who are not from the bio background, like biology background, who doesn't know the biological terms? So I can explain the biological terms also. Okay, I see Mayank raised hand. So I asked them, I want people who are not from the bio background or who doesn't have any biological foundation, who cannot understand the biology terms, to raise their hands. Okay, I expect no one. Okay, I'll continue with the sequence analysis now. So sequence analysis is a process of subjecting a DNA, RNA, or peptide sequence. So these are the biological molecules to any of the wide range of analytical methods in order to understand its features, function, structure, or evolution. The methodologies used include sequence alignment, like I mentioned, search against the biological databases, and others. So, since there is a development of high throughput production methods of genes and protein sequences, so we see there are number of new sequences has been derived, new proteins, new DNA sequences has been sequenced. So, so such a sequences doesn't itself give an understanding to any scientist or any student. So, to increase the understanding of the scientist or the people who are using it, bioinformatics plays a crucial role. However, so we have a lot of sequences. Comparing these new sequences to those with known functions is a key way of understanding the biology of an organism. So here, when we have a new sequence, comparing these new sequences to those with a known function can give us some understanding about the new sequence. Thus, sequence analysis can be used to assign function to genes and proteins by the study of similarities between the compared sequences. Nowadays, there are many tools and techniques that provide the sequence comparisons, like that is sequence comparison, nothing but sequence alignment, and analyze the alignment product to understand its biology. Okay. Sequence analysis includes a wide range of relevant topics. The first one is comparison of sequences in order to find similarity, often to infer if they are related, that is, they are homologous or not. Second thing is identification of intrinsic features of the sequence, such as active sites, or where is the post-translational modification sites, gene structures, where are the open reading frames, distribution of introns and exons, etc. The third thing is identification of sequence differences and variations, such as point mutations or nucleotide, single nucleotide polymorphisms, in order to get a genetic marker or a disease conditions. Next thing is revealing the evolution and genetic diversity of sequences and organisms. Similarly, in other streams of education, in chemistry, sequence analysis comprises techniques used to determine the sequence of a polymer formed of several monomers. In molecular biology and genetics, we call same process called as sequencing. Okay, let me start with the basic molecule DNA. So, wherein this exploration could be helpful. Yeah, just excuse me for a second. I have a question. I'll answer the question first. Yeah, these recorded sessions, we are recording the session, and these recorded sessions will be available through our YouTube channel, where we'll be sharing within a day or two. And whatever the other my co-students, they're taking the classes, they'll also share their recordings with you. Okay, let me start with the basic biomolecule DNA. So we all know DNA is a polymer composed of two polynucleotide chains which coil around each other to form a double helix, as shown here. So this polymer is tend to carry the genetic instructions for development, functioning, reproduction of all known organisms. And broadly, these are called as nucleic acids, which include DNA and RNA. So alongside proteins, lipids, and carbohydrates, nucleic acids are also one of the four major types of macromolecules that are essential for all known forms of life. So coming to the DNA, in DNA, two strands are known as polynucleotide strands. Here you can see one strand is running from 5' to 3', and other strand is running from 3' to 5'. 5', and these strands are made up of a monomeric units called as nucleotides. These monomeric units form a chain called as polynucleotide chain, or single strand of a DNA. So each nucleotide is composed of four nitrogen containing nucleobases, such as cytosine, guanine, adenine, and thymine. So they are shortly represented as adenine as A, cytosine as C, guanine as G, and thymine as T. So these nucleotides are joined to one another in a chain by covalent bond known as a phosphodiester linkage between the sugar of one nucleotide and phosphate of the next, resulting in an alternate sugar phosphate backbone. The nitrogenous bases of the two separate polynucleotide strains are bound together according to the base pairing rules. So what are the base pairing rules? So whether adenine is always interacts with the thymine and guanine is always interacts with the cytosine and vice versa. So with hydrogen bonds. So these interactions make the double-stranded DNA. And these complementary bases or these nitrogenous bases are divided into the two types, pyrimidines and purines. In DNA, the pyrimidines are thymine and cytosine, in purines, adenine and guanine. Can anyone tell me what is the difference comes between DNA and RNA? You can use chat box.
In DNA, that is the deoxyribose sugar, and RNA it's a ribose sugar.
Okay, I asked in terms of pyrimidines and purines, what changes we have. That, yeah, I can get the
Okay,
answer.
it's uracil and thymine. Yes, sir.
Yeah, that's correct. Thank you for answering. So, therefore, to conclude, any DNA strand normally has one end at which there is phosphate group attached to the 5' carbon of the ribose, that is 5' phosphoryl, what we call, and other end. Yeah, all the answers in the chat box are correct. And other end at which there is a free hydroxyl group attached to the 3' carbon of a ribose, where we call 3' hydroxyl. Next, coming with the largest biomolecules are ever studied are the proteins. So proteins are the large biomolecules and macromolecules that comprise one or more long chains of amino acid residues. Before there are polymers of nucleotides, here there are polymers of amino acids. So proteins perform a vast array of functions within the organisms, including catalyzing metabolic reactions, DNA replication, responding to stimuli, providing structure to cells and organisms, and transporting molecules from one location to other. So proteins differ from one another primarily in the sequence of amino acids, which is detected by the nucleotide sequence of their genes, which usually results in protein folding into a specific 3D structure that determines its activity. Coming to the protein, sorry, I still see people joining, so I have to admit them. Sorry for the disturbance. A linear chain of amino acid residues is called as a polypeptide. So polypeptide is formed by a peptide bond. Here in the image you see two amino acids are joined by a peptide bond by the loss of water molecule. So generally short polypeptides, so short stretch, the 20 to 30 stretch of amino acid in a polymer form are called as a peptides. The individual amino acid residues, as I told, they are bound by a peptide bond. Yeah. Next thing, let me start with the sequence analysis. Before that, most of us could have been ever written a secret message to one of our friends, thinking that no one should understand that. So wherein we will be replacing some numbers with the alphabets or the alphabets with the numbers. So there will be particular rules of order so that you and your friend could only understand the secret code. Similarly, there is a secret code or a decoding happens from a nucleotide to a protein, from a DNA level to a protein sequence level. So decoding messages is also a key step in gene expression, in which information from a gene is read out to build a protein, which allows DNA and RNA sequences to be decoded into amino acids of a protein. Uh, can you guys again please mention me what are the two important steps that we come across before proteins are produced or protein expression or proteins to be formed?
Transcription and translation, sir.
Replication, transcription.
Replication is to increase number of copies of DNA. So the two important steps in protein formation will be the thank you for your answers. Translation, yeah, transcription and translation. So wherein in transcription the DNA sequence of a gene is rewritten in RNA. In eukaryotes, the RNA must go to the additional processing step. So this the broad is a DNA is undergoes transcription to form an RNA, and RNA undergoes a translation to form a protein. But there are set of decoding things to happen in translation step. So these step will be codons. Cell decode mRNAs by reading their nucleotides in group of 3. In a group of 3, they are called as codons. That is, when we have an mRNA sequence, this, considering 3 nucleotides at a time is a codon. So where I'll be explaining more details about codon and we'll be solving some problems based on these codons today. So most codons specify an amino acid, and in codons thing, so this is the codon table, sometimes we call this as a genetic code also. When you observe this table carefully, apart from the regular amino acids, we have 3 stop codons which mark the end of a protein. So these codons in mRNA, so when we have a mRNA, set of 3 are considered, and these codons in mRNA are read during the translation, beginning with a start codon and continue until a stop codon is reached, where when we encounter a start codon, this translation is start, and when we encounter a stop codon, translation is stop. And how these are, so initially I mentioned we have 5' to 3' and 3' to 5' strands of a DNA, how they are read. So these mRNA codons are read from 5' to 3', and they specify order of amino acids in N-terminus. Okay, when we here is the codon table that I given here. When we notice that many amino acids are represented at table more than by one codon. For instance, can anyone tell how many ways a leucine can be coded, or number of codons that code for leucine?
Six, sir.
Yes. Yeah. Yeah, there are 6 ways. So when you observe the first box, we see like the last 2 codes for the leucine, and in the second box, that starts with a C, where you see the 4 of them will be coding for the leucine. An important point about genetic code is that this is universal. That is, with the minor exceptions, virtually all species from bacteria to us use the genetic code as shown in the table. So now we'll be learning about the one more concept also, that is reading frames. To reliably get mRNA from a protein, we need one more concept, that of reading frame. Reading frame determines how mRNA sequence is divided up into codons during translation. This is a pretty abstract concept, so let's look into the example and understand here. So this is a one strand, that is 5' to 3', we call it as a forward strand, and this strand has 3 frames of reading. That means this strand can be converted into, from this strand we can extract 3 types of 3 combinations of 3 different ways of codon combinations, where we start considering the first element wherein we leave the first element in the second, and in the third case we leave first two and consider the third one as a first element and continue. In all these, the start codon is the key signal because translation begins at the start codon and continues in successive groups of 3. The portion of the start codon ensures that mRNA is read in the correct frame. Like in the example, you can see a start codon is mentioned in the frame 3. Okay, and how to identify these different frames and backward, what happens when we have a backward frame and all, we'll be learning today using a emboss tutorial at the last. Coming to the sequence based features, here I have mentioned a basic sequence based features, that is a GC content and AT content. So generally GC content is usually expressed as a percentage value, that is G plus C, the number of G plus number of C divided by whole number of nucleotides in that into multiple by 100, and similarly AT content. And other sequence based features that are discussed by Sir during this week's lecture, I'll be explaining them using an example problems. So the final lecture corresponds to the biological databases. So these are the databases consisting of biological data like protein sequencing, molecular structure, DNA sequences, etc. in an organized format. Can anyone tell me what is a role or a need of a database or general features of a database? Okay, the general role of a database, like the major role we know is databases are used to, retrieve, store. Can updating a data be a feature or a characteristic of a database? Yeah, to organize, that's correct. Yeah, generally the major roles are like update, retrieve, insert. So several computer tools are there to manipulate the biological data, like update, delete, insert, that we got a databases. So scientists or researchers or students from all over the world enter their experimental data and results in biological databases so that it is available to a wider audience. And you see, the experiments are so scattered, majorly in the field of biology, their experimental data is so scattered. And these data is collected and organized into several databases which made a wider universe of profit to a larger target audience. And these biological databases what are created are free to use and contain huge collection of variety of biological data. So when we understand what are the uses of biological databases, it helps researchers to study the available data and form new thesis or sorry for the mistake over there. Secondly, it helps people to understand the concepts of biological phenomena, and this also acts as storage information. And coming to the types of biological databases, basically there are 3 types of biological databases, that is primary database, secondary database, and composite database. Now you may have encountered several names like PDB, NCBI, DDBJ, EMBL. So they fall into any of these categories. So I'll be discussing each of these databases detailedly. Coming to the primary databases, so they can be also called as RPL database since it archives the experimental results submitted by the scientist. This database is populated, primary database is populated with experimentally derived data like genome sequence, macromolecular structure, and etc. The data here remains uncurated. So what do you guys mean by uncurated? Means no modifications are performed over the data. So there won't be any check of the data, whatever the researcher or the scientist inputs, that is given as a raw format. And these primary databases also obtains unique data from laboratories, and these data are made to accessible to normal users without any charge. The data are given with accession number. So how can we identify different data? So each data is given as a unique identifier or accession numbers where they are entered into the database. Just like a PDB ID, this PDB or your name. So this name corresponds, like my name is Ramakrishna, so that is given as a unique identifier to me. Or your roll number in your college, where it starts with your branch, year, identifier to your course, like that. So the examples of primary database will be nucleic acid databases like GenBank and DDBJ, DGB, and the protein databases are PDB, SwissProt, Protein Information Resource. Secondary databases. The data stored in these types of secondary databases are analyzed result of a primary database. So when you curate or analyze the result of a primary database, those kinds of data are stored in a secondary database. So computational algorithms are applied to the primary database, and meaningful and informative information is stored inside the secondary database. In secondary database, the data here are highly curated, that is processing the data before it is presented in the database. So what is in the primary database? The data is uncurated. That's a raw format, whatever the research or experimental people put in there. A secondary database is better and contains more valuable knowledge when compared to the primary database because it's a curated and it's validated by several computational algorithms and methods. So the examples of secondary database are Interpro, which has information about protein families, motifs and domains. Uniprot knowledge database, which has sequence and functional information on protein. And the third type of is composite or derived databases. The data entered in these types of databases are first compared and then filtered based on desired criteria. That is, they are derived based on a filter. Yeah, so this comes to the as I mentioned you, further that the biological databases were also classified based on structure, sequence, and other function. Where you see in structure, the widely used PDB comes and sequence databases, SwissProt and GenBank DDBJ, which all these are discussed by sir. So next I'll be moving into the quiz. So before moving into the quiz, I would like to take any questions you have till now what I have explained. So the next questions are going to be based on what I explained till here. So I'll be providing the solutions also. Okay. Okay. Before starting into the question. I have already launched the first question for today, that is, can you calculate the average hydrogen bonding energy for the given sequence ATA-GC? And this same question could be found in the Google Meet polls, and people who are unable to access the Google Meet polls can provide their answer using chat also. And these are not, this is for practice purpose. This doesn't contain any marks, so you can freely answer your questions. Even if it is wrong, no issue, I'll be discussing the answer. Yeah, I see people answering. Yeah, someone has raised their hands. Do they have anything to ask? Yeah, again I'm repeating. We are starting with the quiz session for today. The first question is available on the screen as well as in the Google polls. Yeah, people can answer it in the polls or in the chat box. I'll wait for a minute and then I will try to solve the answer. Yeah, I see some answers. Yeah, okay, I'll move on to the solution. Okay, give me a second. Yeah, so first we need to consider what is the given sequence here. So the given sequence here is AT, A, G, C, and for the in the options we have given information about dinucleotides, that means 2, so X comma Y for example. So the first step is we need to obtain the possible dinucleotides from this given sequence. So we need to construct all the possible combinations in a sequential order. So that what in biological terms is overlapping dinucleotides. So first comes AT. Since it is overlapping, I am taking T again and writing the next element A. Again I am taking A and writing the next element G. Next I am taking G and writing the next element C. So when we take a sequence of length 5, how many dinucleotides we got? 4. That means in case of dinucleotides, number of dinucleotides will be n minus 1, where n is the length of the sequence. Okay, now let us solve with the problem. Now we have 4 combinations. So what is the formula? AT plus TA plus AG plus GC divided by number. So that is n minus 1, or in other terms we can write it as 4. So we have 4 pairs. Okay, then if we take the value and calculate, we'll get minus 35 divided by 4, which equals to minus 8.75. Am I clear here? Yeah, I see most of the people gave correct answer. Anyone having any doubt in this, how we got the values? Thank you for that. Now we'll start with the second question as well. The answer is minus 8.75. And second question is number of overlapping trinucleotide units obtained for a DNA sequence of length 20 nucleotides. Once again, I'm repeating the question. The question is available in the Google polls on the screen as well. So, number of overlapping trinucleotide units that we can obtain if we have a DNA sequence of length 20 nucleotides. Yes. Someone has raised the hand. Okay. Initially, we have solved for a sequence where dinucleotides. So, number of dinucleotides are denoted by number of dinucleotides are denoted by n minus 1. So let's try with an example for trinucleotides, how we can represent. So I'm taking 80 GC here. So the first set will be 80A. Next, since we need to take the overlapping, I'm taking this second overlapping element and adding it to a new element TAA. Sorry, TAT. And next I'm taking ATG. And I'm taking TCG. So using a sequence of n equals to 6, I got 4. So the possible formula could be n minus 2. So here we have asked for 20, n equals to 20. That means the final answer will be 20 minus 2, that equals to 18. The answer is 18. So when we are considering a dinucleotide overlapping, so number of overlapping dinucleotides we get is n minus 1, and number of overlapping trinucleotides we get is n minus 2, where n is the DNA sequence length or number of nucleotides present. Okay, let's start with the third question. The third question is about if a strand of DNA is 10% adenine, then what percent of guanine is present? So we are solving a third question. If a strand of DNA is 10% adenine, then what percent of someone asked me regarding the previous question. So where in the previous question we are trying to identify, given a sequence of 20 size, how many possible overlapping trinucleotides we can extract. So the formula is n minus 2. So n is 20 So 20 minus 2 is 18. Going to the solution. So before I have mentioned about DNA base pairing. So A always base pairs with T and G always base pairs with C. Before moving into this explanation, I'll be explaining about yellow. So this is called as a Chargaff rule. So in the 1950s, a biochemist named Erwin Chargaff discovered that the amount of nitrogenous bases, that is adenine, cytosine, guanine, were not found in equal quantities. That is, he didn't see all of them are equal. There is varying quantities or concentrations among these four nitrogenous bases. So based on this, and further he also observed that amount of A is always equal to the amount of T, and amount of C is always equals to the amount of G. Based on this, he has framed some rules. Those are called as Chargaff rules, which states that DNA of any species or any organism, the amount of guanine should be equal to the amount of cytosine, and amount of adenine should be equal to the amount of thymine. Further, a 1:1 cyclomatic ratio of purine and pyrimidine bases, that is A plus G equals to T plus C, should exist. And he also mentioned two rules, that is first parity rule and second parity rule. So the first rule holds that double-stranded DNA molecule globally has percentage pair equality, that is percentage of A equals to percentage of T, and percentage of G is equals to the percentage of C. Okay. So according to this rule, so the percentage of A should be equal to T, and percentage of G should be equal to T. And given we have this is 10%, both equals to. So these comprise totally 20%. And
Sir,
yeah, yes,
uh,
please.
uh, just want to just tell one thing. The question is wrong. I think the question is wrong because from one single strand of DNA, we cannot tell how much G is there. So the question should be in a double stranded DNA.
Right.
So the question is like if a strand of DNA is has 10% adenine, so we cannot tell that. So I know the answer should be 40%, but I voted wrongly because I was not sure if the question is like one strand of DNA or two strands of DNA.
Sorry. Thanks for the pointing out the mistake. I'll change the question. It's a double strand only. So and next, when we take, the remaining is 80%. So this 80% is equally shared between G and C. So we'll get they both equals to the 40% in a double strand. So the answer is D, 40%. The next question is, if a DNA sequence is given, so what is the possible hydrogen bonds? So consider this is single strand sequence. So the possible hydrogen bonds it can form with the other strand, that is base pairing. So how many total hydrogen bonds there would be formed? this is the given sequence. So we know the base pairing between A and T and G and C has specific number of hydrogens bond between them. So when we consider C, it forms 3 hydrogen bond with G, or when we consider G, it forms 3 hydrogen bonds with C. And when we consider A, it forms 2 hydrogen bonds with T, and when we consider T, it forms with A. So this is the base pairing principle. So based on this, C can form with this complementary base pair G 3 hydrogen bonds, where G with its complementary base pair C, it can form 3. T with A can form 2. T again can form 2. T again will form 2. G will form 3 with C. A with T it will form 2, and C it will form 3. So when you add up all this, you'll get 20. So 20 is the correct answer. Yeah, most of them gave the correct answer. 20. So this is how we see the backbone and the base pairing and the hydrogen bonds you see among A and T and G and C. Next question is the what should be the complementary strand for the given sequence. So the next question is, I have given you a 3 dash to 5 dash sequence. Can you find what would be the complementary sequence of it? Here the given sequence is CG, T, 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 G, A, C, and this is T. So the complementary base pair for C will be, oh, sorry. So the given sequence here is AT, G, G, C, T, T, G, A. So the complementary base pair for A will be T, for T will be A, and for the G will be C, C, G, A, C, T. Since this is 5 dash, it will be 3 dash, 5 dash. So the correct answer will be B. Okay, moving on to the next question. So where we have given number of nucleotides or how many nucleotides are needed to code for a protein that is 100 amino acids long. So this is the question that is related to the codon or decoding steps, or codon table related steps. So if you have a 100 protein amino acid, So what is the how many nucleotides are required for to generate a protein that is a length of 100 amino acids? This question I got all the correct answers. Everyone answered correctly. So as I mentioned, 3 nucleotides were together called as codons, and these code for one amino acid. So when we have 100 amino acids, so they are coded by 3 into 100, that is 300 nucleotides. As one amino acid codon, one amino acid has a codon corresponding with the 3 nucleotides. So 100 amino acids will have 300. So the correct answer is 300. And how this translation will happen and all, we'll see and we'll solve. And the next question is whether it is easy to break a bond between A and T, or to break a bond between G and C. It's a true or false question. So is it easy to break the bond between A and T than between G and C? If you think it is easy, answer true. If you don't think the statement is correct, answer it as false. Can someone explain the answer and why do you feel it?
Sir, actually, between A and T, there are two bonds, two hydrogen bonds, and between G and C, there are three hydrogen bonds. So it is easier to break two hydrogen bonds than three hydrogen bonds. So the answer will be true.
Thank you. And whoever answers in chat, they are correct. Yeah, it's true. The same reason. The A and T holds a two hydrogen bonds between them, and G and C has three hydrogen bonds. So breaking two is easy than three. So next question is related to the emboss. I think whoever has watched the videos can answer this question. Which among the following emboss is used for finding the complementary strand for a sequence? So these are the different emboss too. We'll also at the last. Sorry, I didn't host. answer is RevSeq, where RevSeq is provided with an option to find the complementary strand as well as the reverse complementary strand. So we'll see how to get a complementary strand and reverse complementary strand in RevSeq today. So next question is DDBJ stands for. I don't have much space in the quiz to load this question, so whoever wants to answer, they can use the chat box to answer. Yeah, the correct answer is DNA Data Bank of Japan. So I forgot to mention it before, so I'll be giving a brief about the DNA Data Bank here. So the DNA Data Bank of Japan, shortly known as DDBJ, is a public database of nucleic sequence established at the National Institute of Genetics (NIG). So this DDBJ has accepting annotated nucleotide sequences, and they issue accession numbers just like recognition numbers, and distributing them in collaboration with GenBank and NCBA. GenBank at NCBA and ENA at EBA, European Bioinformatics Nations. So this is a collaborative framework, so where DDBJ, GenBank and NCBA are a collaborative framework where they share information among themselves. This collaborative framework is known as International Nucleotide Sequence Database. Yeah. Next question is regarding again codons. So which of the following amino acid has only one codon? So the question is, which of the following amino acids is coded by only one codon? As I mentioned, codons in mRNA are read during translation beginning with a start and continuing until a stop codon is reached. So in this codon, we have 20 amino acids that are coded, and we have 64 codon combinations. Great, all the answers are correct. So answer is B. I'll show you a codon table here. So given the given options, tryptophan is correct as it is only coded by one codon, and when we consider the other, the start codon is also coded in one way. Methionine also has only one codon, but in the options methionine is not given, so tryptophan is the correct answer. Next question is, GenBank contains information on. So what kind of sequence information or the macromolecule information that GenBank holds? Yeah. While you answer the question, so I'll explain about the GenBank. Now recently this is one of the fast growing repositories and has a flat file structure, and this is also an open access sequence database, and it coordinates with the individual laboratories and other sequence databases like EMBL and DDBJ. Here GenBank is a nucleic acid sequence database, and this is so let's have some quiz questions where these are the numerical answer type. So the first question is, how many different types of nucleotides are found in RNA? Nucleotides or nitrogen bases that are found in RNA. AUGC. Okay, you have given the name. Yeah, in RNA we find 4. So instead of 4 types, correct? The second question is how many types of amino acids are found in proteins? So I expect, I'm thinking questions are very easy. People are answering so fast. Yes. 21. Okay. Yeah. In general, we see 20 amino acids that are found in proteins. There are exceptions. There are some leucine and other things, but we are not considering them here. The second question is how many nucleotides are needed to code for single amino acid? Let's not waste much time on these easy questions. So next question is different codons are there in genetic codon table. Okay, I've seen someone mentioning 61. I expect them to avoid stop codons. Okay. Yeah. In a codon table we have 64, including that code for the stop codon. Next, how many codons code for amino acid? That's 61. The rest 3 are the stop codons that dictate the stop of synthesis for a protein sequence. So before we go into emboss, let's try manually to solve these questions, and then we'll try to solve these questions using emboss also. So the first question is find the complementary strand for the sequence. I know it's time taking for you people to type a long sequence, but this would be a good practice, like solving manually and then experiencing the emboss tool is a good practice. So I'll wait for a couple more minutes and you can guys answer this question. So the first question is I have given a strand of DNA and we have to find the complementary strand. Yes, I'll wait some more time. Okay, all the answers are correct. Okay, now I'll provide you the answer, but we'll check whether this answer is correct or wrong in the imposter. Next question is to find the reverse complementary strand for the sequence. I think that this could be an easy solution for the people who solved the first one. So finally is I have given you a RNA sequence. Can you translate it to a protein sequence? So consider this as a forward frame and first give me a first frame. So I initially showed you frame 1, frame 2 and frame 3. No? We'll solve frame by frame, that is frame 1, frame 2, frame 3. So first provide me with a frame 1 amino acid sequence. In order to solve this, you guys can take a help of codon table you want me to show. Okay, no one has yet solved for frame 1 also. Okay, frame 1 will be methionine. Yeah, when you consider the first one here is AUG, that codes. Yeah, that's correct. Okay, let's move on to the frame 2. So when you are making for the frame 2, we'll remove the first element and we build the 3 combinations from U. So the first combination will be UGU. So we'll exclude A from in second frame. Yes. Yeah, that's correct. And when we are solving for the frame 3, we'll try to avoid first 2 elements and take and consider the third element. So here we'll be trying to avoid A and U. So our first codon will be GCU. Sorry, GUC. We'll try to avoid A and U. So third element is G. G, U, C is our first codon. And for your information, the recording and the slides we have discussed and with the solutions will be provided to you in a day or two. Guys, keep on doing it. Yeah, the frame 3 is also correct. Now we'll try to solve the same using the emboss tool. So in emboss tool, okay, I'll share my screen again. I hope my emboss tool is visible. So this is the interface of how emboss looks, where you can see several information about emboss on the left hand top, and even the documentation is provided. So this documentation has information about the different tools and the roles and how to tutorials for them. So first we need to find the complementary sequence. So what is the tool that the sir has mentioned in the class for the complementary sequence? Even we solved it a question today also. Yes, yeah, it's RevSeq. So on the left hand side you can find the go to option where you can enter the name RevSeq, and you can find RevSeq at the bottom. So when you open the RevSeq, uh, looks like this. So where you can browse files for input, or we can paste the file. So I'm pasting the file. So this is what our input. No need to mention the 5-N and Preethram, just input the sequence. So the tool for finding a complementary sequence is a RevSeq in Emboss. So I have searched for RevSeq sequence in the go to option provided by Emboss. Then I double clicked on it to open. Once I double click, the RevSeq provides me option to input a file or to input or paste the sequence. I'm selecting an option to paste the sequence. Once I selected the sequence, I have pasted the sequence, I'll go for the advanced option. So in the advanced option it tells whether you want to print a reverse sequence, and it will ask whether you want to give a complementary sequence, and you have to have a tag output. So now I'm selecting only the complement sequence. I'm not selecting the reverse of the output, and I'll click go. You can see a output is generated here. So we can, this is a complementary strand for the given input. Now this could be your practice session, or you can have hands-on with a number of sequences you have. Let us check. Now this is given sequence. Let us check what happens if I select reverse sequence. I selected the reverse sequence and click glue. So we can see we have got the reverse complementary strand. So the RevSeq can use to find the complement strand as well as the reverse complement strand as well. Okay, our next question is finding this one. I'm copying this. Can anyone know what is the tool to translate a RNA to protein? Yes, yes, it is Transseq. So similarly, I'm going to the GoTo to search for the Transseq. So the Transseq appears at the bottom of the GoTo window. Double click to open. Again, I'm selecting a paste option to paste the input file. I'm pasting the RNA sequence. Now, Transseq gives us many options like frames to translate. So we have discussed that forward strand will have 3 frames and backward strand will also have 3 frames. So overall, we'll have 6 frames. So here the option is given to get a 1 frame or only the second frame, only the third frame, or only the set of 3 forward frames, or we can get all 6 frames. So I'll select all 6 frames and I'll click go. So you can see all the 6 frames are here. You guys may not see the output on my page, but once you click, you can see. So this brings us to the end of the session. So any questions or any doubts? Yeah, I'll add how to install Emboss and for different Linux version and Windows version, how to install, where to download. Those instructions I'll add and upload the files, upload the PPT, I'll edit the PPT and add. So where you can play with Emboss. Emboss has several other tools also. These tools can be found in the documentation part of the Emboss, and detailed explanations along with the solved examples, everything has been given there. Any doubts you have, or you are free to leave. And people who have joined rate recently wants to know anything about the session, they can also ask the question.
Sir, can you explain the just overall brief what happened today, sir?
Yeah, I'll be explaining like in a couple of minutes. If anyone is having any questions, I'll be taking them and then explaining it. Yeah, if you have no questions, you are free to leave. Just I'll be giving summary, a brief summary to others who joined late, and then we'll be winding off the session.
Yes.
And the recording and PPT will be available within a day or two. We'll be uploading it and we'll be sending you a mail.
Where will be finding the recorded PPT, sir?
In the same announcements portal where you have found the link for the today's session, you will be finding in announcements portal of the
Okay,
Swayam
okay,
page.
okay.
Hello.
Yes.
Good evening, sir.
Yeah, good evening.
So, what about the remaining sessions we missed? No, actually, we are listen those classes.
You have attended those classes or not?
2 days classes,
Yes,
live sessions.
this is the on week 1 you'll have 4 people teaching, like 4 sessions going on. This is the third one. Tomorrow you'll be having one more taught by other one. It's like, see, all the sessions recordings will be made available to you.
And where do you listen, sir?
In the
Recording.
announcements portal of Swayam page, when you see announcements portal, we'll be uploading it.
I have uploaded. Okay.
Yeah,
Recording
yeah,
videos, sir.
yeah, we'll be doing it, uploading in the Swayam portal.
Okay, sir. Thank you, sir.
Yeah, okay. For those who have come late, so today we started this session with an summary to the week 1's course, what sir has taught, Michael Romeo sir has taught. So where I started with the introduction to bioinformatics and how bioinformatics is useful and how the bioinformatics is used to solve the complexities. Then I started with the explanation to the basic biomolecules such as DNA and protein. Then we entered into sequence analysis part where we started learning about what's a codon table, genetic code, and how to translate and different frames that we encounter during the translation step. Then we discussed about the different biological databases that are primary databases, secondary databases, and derived databases, where the information is already available in the slides. I'll be providing you these slides only. Then we solved some questions based on the discussions in the first half, and those questions with solutions and detailed answers will also be provided. Particularly if you want to know about any of these concepts, I'll be explaining it again. Who joined late? And simultaneously, Friday, Saturday, and Tuesday, other sessions will also be going. Most or less, we'll be discussing about the same, but do try to attend those sessions also in your free time. If no questions, can I wind up?
Every Friday and Saturday, sir, the sessions are going on.
Friday, Saturday, Monday, Tuesday.
The timing is 6 to 8, sir, or any of the timings are changing?
Yeah, you can find them in announcement portal. I'm not sure about the exact timings, but each session will be more than 1 hour. And in the later session, since this is the first session today I'm taking, so this is very brief. In later sessions, once your assignment deadline is over, we'll also try to help you in solving the assignment problems. Before deadline, we are not supposed to solve. Once assignment 1 deadline is over, I'll be taking up the assignment 1 explanations, detailed solutions will be provided, so which will be helpful for your exam preparation or for gate preparation, anything. This is the first session. This will be very brief and short.